You are welcome to Edexcel IGCSE Biology class. Uh, this is Ajiro Tech. This is where we'll be breaking IGCSE Biology, be breaking it down for you to understand. So this is Edexcel IGCSE November. That is what we'll be shooting. So I seek your attention as we continue to this question. So this is paper 1, as I've said. It's saying the mixture of the multiple choice. So let's go straight to the point. Now, question 1. An amoeba is a microscopic single-celled organism that lives in pond water. So the diagram shows an amoeba with some structured. So, and you can as well see this structure. This is a structure of amoeba. You can see line X and Y. Line is showing you the size. So the question on A says that each structure is the nucleus. As you have known that amoeba is animal cell. There is nucleus, cytoplasm, cell membrane. If you look critically down into this cell, discover that nucleus are always centrally in prokaryotic, eukaryotic cell, especially the animal cell. In plant cell, the nucleus is not centrally placed. It is the vacuole centrally placed. In plant cell. But in animal cell, like these phylum protozoa, either is a protozoan. It is a protozoan. So the nucleus is centrally placed. And a body that is centrally placed, you can see here at the center. So this is the nucleus. Apart from being centrally placed, what can you still use to identify? Let's say not like nucleus in another cell that is not identified. Apart from being centrally placed, what characteristic to identify? One of the properties or characteristics that you can use to identify nucleus is that it is densely colored. It is densely colored. That is the reason why it is visibly spotted. It is densely colored. It has the ability of absorbing a lot of dye or a stain that is used to stain the cell to be viewed on. So any structure in a cell that is densely stained and absorb a lot of stain, that is the nucleus. So you can see S. So the answer is D. I'm going to tick this to tick it. So the answer is D. And because the structure is S. So let's go to the second question. On figure 2 is still about the same amoeba. Which structure is the cytoplasm? Structure is the cytoplasm. The cytoplasm is jelly-like in nature, and it looks like a fluid that flows. So cytoplasm flows. In fact, in animals like the flowing of cytoplasm results into the movement. Yes, we know that amoeba move specialized structure called pseudopodia. Pseudopodia is used by amoeba to move. But at the same time, cyclosis, yes, that is the term used for it. Cyclosis. Cyclosis is known as cytoplasmic streaming. Cytoplasmic streaming. This cytoplasmic streaming means the flowing of the cytoplasm. So the movement of the pseudopodia alone cannot properly propel amoeba through its aquatic environment. But by the help of the movement of the cytoplasm, which is known as the cytoplasmic streaming, or the process called cyclosis, it helps in the amoeba entire cell. So apart from that, cytoplasm is also the center for chemical reaction. It is the center for. So the structure that suits the description that I've just given is this. This is a site. So let's look at the structure that is in structure Q. So structure Q is the cytoplasm. This where chemical reaction of the cytoplasm also helps the amoeba through. So the answer is B, which is Q. Size Q. So if you look at the options, you tick this box. Right, so let's move to the next question. Next question is still about so the question now says, which structure contains chromosome? Yes. Remember that in our first question, we asked the structure that is labeled as nucleus. And we know that chromosome can be found. And you remember I said something in one this question that absorb a lot of stains or a lot of. There is a structure in the nucleus that makes it absorb a lot of, and that structure is the chromosome. In one of the videos that I've made some times ago, I explained this chromosome as a structure that absorbs a lot of dye. It is used in staining cell. So I've told you that chromosome comes from the word, and chrome means color, then some means body. So chromosome means colored body, meaning that it is a structure that is present in the nucleus that absorbs a lot of dyes or the body. And when nucleus is thin, to be more, it will be obvious, to be visible because of the absorption of dye that is used in staining cell. So the structure that contains the chromosome is the nucleus, which is structure S, and that is T, which is this box. So we are going to think this box. So next question. Next question still on question 1. The magnification of the diagram is times 65. Calculate the actual length of the amoeba X to Y. Yes, it's always difficult to calculate magnification from screen. I'm going to base my on assumption. So what you need to do is just to follow the similar step that you need. The same step to use for you is the same step you should use. Don't mind the value. Though there are some fixed value that has been given to us in the question, such as magnification times 65. That one is not changing. It's a fixed value that has been given in the question already. So calculate the actual length. So we are calculating the actual length. So this is what we are calculating. We have been given magnification already. So don't forget that when you are calculating magnification, there are 3 parameters that you need to calculate your magnification. The first thing that you need, in no particular order, because you may be asked to calculate image size in one question. In the other question, you may be asked to calculate magnification. In the other question, you may be asked to calculate the actual length. So what I'm about to list is in no order. So you need your magnification value, So you need your magnification value. Another thing that you need your actual length. That one as well. Then the third thing that you need is the image size or image length. So these are the three things that you need. Now, out of these three things, you must be given two. In the question, like for example, in this question, we have been given magnification. And they ask us to calculate actual length. Image length or image size can be gotten from image that you are in. So what you are measuring is this your line X and Y. And you have already been given scale, a conversion scale. Maybe for example, you don't know how to memorize conversion. So you have made your conversion to be very simple. So ordinarily, as a biology student, you should be able to know. They have made the question to be simpler to know. So that is 1,000 micrometer is equal to 1 millimeter is divided. That is what this measurement is telling you. So you have X to Y line. So what you need to do is to get because your magnification has been given, to get your image length, you have to take your ruler and measure this line X to Y. So it depends on the type of ruler that you're using. If you are using centimeter ruler, you can measure in centimeter and then convert to millimeter or micrometer as the instruction being fit or as the instruction states. Let's look at what the instruction states. The instruction wants us to get an answer in micrometer. So if your ruler is not having millimeter, if your ruler is only having centimeter and inches, I will advise you to measure in centimeter, then multiply by 10,000. Whatsoever your value is, multiply it by 10, so that you can get your reading straight in mind. So I believe you can understand what I'm saying. So you go straight to micrometer instead of multiplying your reading by 10 to get your millimeter, and you multiply again by 1,000 get your reading in mind. Just multiply straight by 10,000. Whatsoever you measure from point X to point Y in centimeter, multiply it by 10,000, get your answer. So let's say I'm using centimeter ruler because it is common. Centimeter ruler is common. So let's say I'm measuring in centimeter, and this line I measured it to be, let's say for example, 6.5 centimeter. 6.5 centimeter. I have told you that the value I'm using here is an assumption value because I cannot be placing ruler on the screen. So, and the zoom level of the screen might not be the same with that of the question paper, physical question paper. That is the reason why I'm saying that as far as this question is concerned, I'm going to use an estimated page size. So you just follow how the calculation is done. So 6.5 centimeter times 10,000 is 10,000. So 6.5 times 10,000 give you, let's move the 10,000 is having how many zeros? It's having 4 zeros. 1, 2, 3. So let me move my decimal points 4 times. The first time, that will give you 65. Just add 3 zeros. That's 65,000. So my image size equals, or image length. I use length. So my image length is going to be 65,000. 65,000 what? My centimeter meter. So now remember this formula that magnification equals to image length actual length, page length over actual, that is the formula. Or we are asked to calculate the actual length. So if I cross multiply, I'm going to have magnification times the actual length equals image length. So don't forget that I'm looking for actual length, which is AL. So I'm going to retain my A. My M will have to go to the other side, which is my magnification. So M, on getting to the other side, will have to divide image length so that my actual length be equal to, actual length will now be equal to image length. So my actual length will now be equal to image length. My image length is 65,000 micron over magnification, which is 65. Yes, times 65, which is 65. Actual length will now be equal to 1. So my actual length will now be 1,000 micron. There is no need to put these units there. There is no need. So your here. So just put 1,000 here. Now that doesn't mean if you check the math screen, it is 1,000 you're going to see there. No, remember, I said that I use estimated using screen, and I'm not using physical paper where I am going to use formula. So this length that is 1,000 micron that you see is an estimation. So what you just need is the calculation that I've done here, how you can go about it. So if you apply the same method in the examination or when you have your paper, get the correct answer. So let's move to the next question. So give 3 differences in structure between amoeba and. Now let us look at the structure of amoeba here. We know that amoeba structure P is actually a ribosome. Yes. Viruses do not have ribosome. Viruses don't have ribosome. Though they have DNAs, they have RNAs, they have nucleic acid, whether deoxyribonucleic acid or ribonucleic acid. They have DNAs. Some are single-strand DNA, some are double-stranded DNA, some are single-strand RNAs, some are double-stranded RNA. I'm talking about viruses. So they have genetic material. That one is cytoplasm. Ribosome, no, they don't. Cytoplasm, no, they don't. So Q, they do not have Q. Cytoplasm, they do not have cytoplasm, do not have ribosome. S, they do not have nucleus. Nucleus, you may be saying, but they have DNA. Why is it that they do not have nucleus? Remember, I said something that nucleus that is bounded by nuclear membrane, they are regarded as the eukaryotic cells. So viruses, they are not bounded by nuclear membrane. So we regard them as organisms that do not have. So I've said they do not have ribosome, they do not have cytoplasm, they do not have nucleus. Yes, they don't have all these structures. Don't have all these structures. Even they don't have vacuum. No vacuum, because all these ones are contractile vacuum. This is a contractile vacuum, even though it's not labeled. It's a contractile vacuum. Viruses don't have it. So there are a lot of structures that amoeba possessed that viruses do not have. So give 3 differences in structure between an amoeba and virus. Between an amoeba virus, yes. So number 1, as we have described up there, that amoeba has a nucleus, because they said 3 differences between amoeba. So amoeba has nucleus, has nucleus. Amoeba is having nucleus. So whatsoever we have written will be towards amoeba. So amoeba is having a nucleus of which viruses do not have. And then ribosome is an organelle. Cytoplasm is also an organelle. So we can say that amoeba has organelles. Do you understand? Amoeba has organelles. Organelles such as ribosomes, cytoplasm, even nucleus is an organelle. Yes, etc. Which cannot be found, which are not found in virus. It cannot virus. So another difference between amoeba and a virus is that amoeba is having cell membrane and viruses do not. Amoeba is having cell membrane. Amoeba is a cell membrane. Which cannot be found, which not found virus. It cannot be found in the virus. So that's that about this question.
In today's world, science isn't just about passing exams. It's about understanding how the world truly works. At Ajiro Tech Tutoring, we help students master the sciences, physics, chemistry, and biology across all major exam boards, AQA, Edexcel, and Cambridge International. With our team of qualified tutors, every session is designed around you, your pace, your goals, your success. We don't just teach, we build confidence, critical thinking, and curiosity. And now, we're proud to say we are officially registered right here in England, in the UK, delivering trusted, high quality online tutoring that meets British educational standard. Whether you're preparing for GCSE, IGCSE, or A levels, Ajiro Tech gives you the tools to achieve your best result from the comfort of your home. Because with the right guidance, science becomes simple, success becomes natural, and learning becomes limitless. Begin your journey today at ajirotech.com or message our sales team info@ajirotech.com or ajirotech@gmail.com. Ajiro Tech Tutoring, where understanding leads to excellence.
So let's look at this. Yes, this is an investigation. A teacher uses this apparatus to investigate the effect of concentration of a substance on the rate of diffusion. Cotton wool with drops of ammonia solution, and you can see the cotton wool here. Then there are damp red litmus paper. Damp red litmus paper means that litmus paper has been soaked in water, and the type of litmus paper is red litmus paper. So we have two types of litmus paper. We have red litmus paper. We have, in this investigation, this is red that is used, and this is a long tube. And one thing I observed in this investigation is that the damp red litmus paper, they are placed at strategic location. The distance between one damp red litmus paper to the other is 4 centimeters apart. So meaning that if a red if first red litmus paper is placed at 4 centimeter, the next one will be placed at 8 centimeter, the next one 12 centimeter, 16 centimeter, 20 centimeter, 24 centimeter, like that. Okay. Now, this is the teacher's method. Let's look at how the teacher carried out that investigation. So, step 1, place 6 small squares of damp red paper in a glass tube at 4 centimeter. Put the glass tube in a film cupboard. Film cupboard. What's film cupboard? Something that we need to understand. The film cupboard is a kind of cupboard that scientists or researchers make use of when they are working with dangerous gases or dangerous chemicals that produce fumes or vapor, corrosive vapor, dangerous vapor, so that the person that is working with such chemical will not be exposed to the vapor. So that is the reason why scientists or researchers or laboratory scientists normally do their work when they are working with that kind of chemical. They pour the chemical, they do whatsoever thing they want to do with the chemical inside this film cupboard. So how does this film cupboard get rid of this dangerous fume? There is an evaporator. There's a special evaporator, special fan attached to the cupboard. So the fan will be working at high speed and the fan will be sucking up the fume and be taking the fume out of the cupboard and out of the environment where the scientist is working, where the laboratory scientist is working, so that the environment will be made safe for the person that is working with chemical. So that is the importance of using film cupboard. So what is this chemical that this investigation is based on that now warrant the use of film cupboard? Is ammonia solution. Ammonia solution always gives off this pungent smell, pungent fume. Yes, because ammonia is poisonous. So that is the reason why film cupboard is advisable to use when someone is working with ammonia solution. So because this diffusion investigation is based on we are using ammonia solution, any ammonia that has been diluted, though it depends on the concentration of, but notwithstanding, if you want to make a particular concentration of ammonia, you make whatsoever concentration you want to make from a concentrated ammonia. So immediately you open the bottle of a concentrated ammonia, the fume will escape. That might be dangerous to the person that is handling it. So that's the reason why film cupboard is highly recommended when this investigation is to be carried out. So add 1 drop of ammonia solution to some cotton wool. Cotton wool is an absorbent material. It absorbs ammonia. The reason why you may be thinking, why can't we just get like a small container, put the ammonia inside and put it inside this long or cylindrical tube that we are using? We can't do like that because we are carrying out an investigation of diffusion. So if we do it like that, ammonia solution, it evaporates. It evaporates. The rate at which it evaporates, if we do not pour it in an absorbent material, the rate at which it evaporates will increase drastically, which may affect our diffusion investigation. So we have to put the ammonia solution inside an absorbent material. Now, this absorbent material, such as cotton wool, will be releasing ammonia gas gradually. Do you understand that? So we cannot just pour ammonia like that in the test tube or put it inside the cover of the test tube. No, we need to use an absorbent material such as cotton wool that will be releasing the ammonia gas gradually. So place the cotton at one end of the glass tube, and you can see this cotton wool at one end. It is placed towards the left of the tube, and then the other end is covered with a rubber bond, that is so that it will not allow the type of ammonia gas that is, being produced or being produced from this end, because they will be moving gradually so that it will not be going out here. So that is the reason why we are adding the rubber bond at the end. So close the tube with two bonds. Yes, this is one bond at this and this is the other bond, so that there will not be any gas escape. So call the time taking. This is very important. Time taking is very important for the ammonia to change the color of each square of the tube. Now this is where we are going. It is very, very much important for you to notice the color change, because the color change and the time it takes for each damned red litmus paper to change to blue is very, very important. Why are the litmus paper, why would they change from red to blue? The reason why they change from red to blue is because ammonia is acidic in nature. So it changes red litmus paper to blue. So that is what will be observed. The color will change from red litmus paper to blue, and from, if it is another substance and not ammonia that is here, it will change blue litmus paper red. It will change blue litmus paper red. So that is what we are looking at here. So now, what it takes, the time it will take for this color to change from red to blue. Now, the reason why the color will change, because you will be thinking that this damp red litmus paper, why must we soak it inside water to make it damp? This is because as ammonia gas is giving off, so that it will dissolve. That's the reason why we have to wet our litmus paper, red litmus paper, with small amounts of water. That small amount of water is, it will give the opportunity the litmus paper to be soaked, and then allow the ammonia gas to dissolve on the red litmus paper, so that it can easily detect the ammonia gas and then change the color to blue. So that is that's that. Then record the time taken for the ammonia to change the color square of the litmus paper. The teacher repeats the method with 3 drops of ammonia solution instead of 1 drop. So we have, according to this investigation, we have 2 independent variables. So just one independent variable. The one independent variable is the number of drops, or let's just say the concentration of ammonia. The concentration of ammonia is the independent variable. But we have 2 types of concentration of ammonia. It is still one independent variable, which is concentration of, but the concentration of ammonia is in 2 ways. One is 1 drop, another one is 3 drops. So in case if we are asked to plot the graph, so they may ask us to plot the graph of the 2 together. So that is a graph with 3 drops and a graph with 1 drop. So state what is meant by the term diffusion. Now, if you are defining diffusion, there are some key words that you must mention if you are defining diffusion. So you will have to say that diffusion, it is the movement, of particles. It is the movement of the particles is the movement of particles. Particles of what? The particles of gas, the particles of the liquid. It is the movement of the particles or molecules from region of the higher concentration, to the region of lower concentration, to the region of concentration. So that is the definition of diffusion. Now, let's apply this definition that we have written. Let's apply it to this investigation and let's see if it relates. It is the movement of particles. Particles of what? Particles of ammonia. Because the ammonia will be evaporating. Ammonia gas, the particles will be evaporating. As it is evaporating, it will be spreading. Now, the first damp red litmus paper is spotted at 4 centimeters. Now, the 4 centimeter damp red litmus paper is the one that will first change to blue. This 24 centimeter at the end will still remain blue, but will still remain red to not change to blue because the particle has not moved. Because this is the cutting wood. This is the area of higher concentration. Higher concentration. Because you have more of ammonia on this side than you have it on this side. So this region is the region of lower concentration. It's the region of lower concentration. Why this region to the left is the region of higher concentration? Because that is where we place our cutting wood. Then the other region here will be at the right hand side, be the region of lower concentration. So the movement of the particle of ammonia from the region of higher concentration to the region of the lower concentration, we can see that it matched the investigation perfectly well. So let's move to the next question. 2B. You can as well see the table. This is after the whole investigation. The teacher has represented his results on this table. So this is results table showing us after the whole investigation. So this table, we have number of drops of ammonia solution, which is our independent, because this 1 and 3 is telling us the concentration. 1 drop is representing lower concentration. While this 3 is representing higher concentration. So the independent variable is that we are varying the concentration. We are varying the concentration. So and you can see on the table, you can see that the time it takes for lower concentration for diffusion to take place in 1 drop of is higher. The time taken is higher. But here in a higher concentration where you have 3 drops, the time taken is lower. So you can see now, if the dependent variable in this investigation, what is a dependent variable? A dependent variable is the variable that is measured in the course of an investigation. Dependent variable is the variable that is measured in an investigation. Yes. So now let's look at this investigation. What are we measuring in this investigation? Or what has the teacher measured in this investigation? You will see that the teacher has measured the time taken for litmus paper to change the color. That is what the teacher has measured. Teacher has measured the time taken for litmus paper to change color. So now give the dependent variable in this investigation. So the dependent variable is the time taking. Litmus to change color seconds. So you might not add this in seconds, you might not add it to it. That's in units, you may not add it to it. But the time taken for litmus to change color is fine. So let's go to the next question. Okay, the next question says we should plot a line graph to show the time taken for the litmus paper at each distance to change color. Use a ruler to join your points in straight line. When you are solving this question, discover that you have to use a ruler to join points with straight line. Use ruler to join your points with straight line. Use a ruler to join your points with straight line. So now draw a separate line for 1 drop ammonia solution and for 3 drops of ammonia solution. So let's do it like that. So this graph is going to have 2 lines. One line represents 1 drop, the other line represents 3 drops. So let us these are graph sheets. So let's draw it up. Now when you want to plot graph, you need to be careful. You need to study the units, the scale that you are going to use. You need to study very well. Now let's look at the scale for time taking for the litmus. Now apart from the scale, what you also need to consider is your dependent and independent. Your independent variable. Now on this graph, your independent variable is your distance. Is your distance in centimeter. So all these 4 centimeter, 8 centimeter, 12 centimeter, 16 centimeter, 20 centimeter, 24 centimeter will all be drawn at your x-axis, while your time will be drawn on the y-axis. Because time is a dependent variable, while distance is your independent based on the investigation that we are working on. So now let's go straight. Let's see how we can partition our graph sheets into specific lines, into specific readings, or into specific scales. So this is the x-axis. Actually, is the x-axis, and this is the y-axis. This is the y-axis. You may not name them x and y because your axis, they have name. So we are going to name our axis first. So, and our axis is, for example, this x-axis now is a distance traveled by, let me check the question again. Place the cutting rule at one end. Okay. Place 6 small squares of downward length in the last 2 at 4 centimeter interval. As a distance between litmus paper. Let's put it that way. Distance between litmus paper. So this is distance. Let me change my pen ink to the one that will be visible. Let me change it to yellow. I think this one is visible enough. So distance between litmus paper. Distance, let me just put litmus. Distance between litmus. It is measured in what in centimeter. So yellow, the yellow color, yellow can show. Let me change back to blue. Let me change back to blue. So now let's begin to mark our distance. So this is 0. The highest reading is actually for the distance is 24. The lowest is 4. The highest is 24. So let's see how we can write this line. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. 24 divided by 12, that's 2. Okay, 24 divided by 12 will equal to 2. So each line we can say 2, 2. So this is 2, this is 4, this is 6, this is 8, this is 10, this is 12, this is 14, this is 16, this is 18, this is 20, this is 22, this is 24. Yeah. So distance between litmus paper, we have done that. Now let's do time taken for litmus paper to change. Time taken for litmus paper to change. Let's name this axis. Time taken for litmus paper to change color. Yes. So now don't forget the units is measured in seconds. This seconds. Let me check the table again. Yes, it's measured in seconds. So just put your S, close the bracket. You've gotten 1 mark like that by properly labeling your axis. So let's now see what we can use. The highest measurement for drop 1 is 38, or 3 drops is 22. So the highest measurement is actually 38. So let's show our graph to 38. Okay. Let me do it like this. Let's say 2 centimeters represent 5 units. So from here to here is 2 centimeters. So let's use it like that. It represents 5 units. So let's see if it's going to work out. So this will be 5. So that 2 holes will be equal to 1 unit. So that this and this will be 1, this and this will be 2, this and this will be 3, this and this will be 4, this and this will be 5. So that's very good. So we have 10 here, we have 15 here, we have 20 here, we have 25 here, we have 30 here, we have 35 here. Don't forget the highest is 38. So we have 40. Let me claim this Y. We don't need it. So we have 40 here. So this is very, very perfect. All right. So now don't forget that we need to have key. Yes, because we are dealing with 2 independent variable values, 1 drop and 3 drops. So let's have key here. Let's have key here. So the key is to let us know the difference between the line of drop 1 and the line of drop 3. 1 drop and 3 drops, so that we will know the difference. So let's use continuous line for drop 1. Continuous line is representing drop 1. Don't let me say drop 1. Let me say 3 drops. Let me play 1 drop. 1 drop. Then broken line. This broken line represents 3 drops. 3 drops. So anywhere you see continuous line, that's 1 drop. Anywhere you see broken line, it is 3 drop. So let's do that of 1 drop first. So at 4 centimeter, we have 8 seconds. At 4 centimeter, we have 8 seconds. 8 will be around this place. You know, this is 5, this is 6, 7, 8. Okay, this is 10. Since this is 10, this will be 9. Let me enlarge it so that we'll be able to see. Yes, enlarge it so that we'll be able to see what we are plotting. So 8, we want to locate 8. So this is 5 here. This and this will be 6. This and this will be 7. This and this will be 8. So here now, so let's mark our points. Yes. So let me use a plus sign. This plus sign to mark. So that's the first point. Let's go up and let's check our table. Let's check our table for the next point. The next point is 8 centimeter to 15. 8 centimeter to 15. Let's go back. 8 to 15. 8 to 15. Up here. This is 15 up here. Now 8. These are 8. So where I'm going to put it? Let's put it here. Yes, that's nice. So let's go again. Let's go again. That's 12. 12 to 20. 12 to 20. 12 to 20. 12 to 20. It's 12 to 20. This is 12 here. Let's trace it to 20. Yes, this. Please, I may not be able to use ruler, but I will try to make my hands be straight and join those points. Anyway, so 14. 14. We don't have 14 here. 12 to 20. 16. Okay, it's 4 centimeter apart. 4 centimeter apart. It's 4 centimeter apart. So all right, it's 4 centimeter apart. So let me so it's 4 centimeter apart. So let me now, this is 16 to 25. 16 to 25. 16 to 25. 16 to 25. Okay, this 16 here. Let me reduce the zoom a little. 16 to 25. Yes, see the 25. So let's trace the 16 up, then to 25. 25. Let's do it here. I always like my graph to be big, to be large. You know, there's beauty than having a jam-packed or squeezed graph that will not be spacious, that you will not be able to see everything clearly. I intentionally make use of everything, all the graph sheets, so that it will be big enough, it will be attractive. Do you understand? So the more you make your graph attractive, not by shading anyway, or by carefully drawing your lines, and I don't even need to draw any straight line. I made use of the line that has been drawn there, and I carefully chose my scale. If you carefully choose your scale and you carefully draw your graph will be neat. The graph will be very, very neat. So let's go straight to the next one, which is 2031. 2031, yes. 20, 31. Okay, it's 20 here. 31, 20, 31, 20, 31. All right. This is 30 up here. This is 30 up here. 31 will be 1, 2. So 31 will be 31. Yeah, that's it. I love that. So then the last one, 24 to 38, 24 to 38, 24, then 38. This is 35. This is 36, 37. This is 35. 1, 2 is 36. 1, 2 is 37. 1, 2 is 38. So this. All right. So Y24 is the last year. So we're going to put it here. This 38. This will be 39. All right. So now, don't mind. Let me draw my line now. I'm done with one draw. So let me draw my line. Please don't mind me if it is not straight. It is not straight. Don't mind. But during the exam, you will have to use ruler. You have to use ruler. I know there is a tool that will help me draw a straight line, but I'm not sure if we have it. For sure. Just let me use my free hand. Just accept it as it is. I'll be careful as possible. It's not passing through. I mean, my hand is not straight enough. Okay. Yeah. So that is for one drop. Now let's do for 3 drop. For 3 drop. That's 4 centimeter 2. 4 centimeter 2. 4 centimeter 2. I'm going to use okay. It's 4 centimeter. Then 2. This is 1, 2. This is 1. This is 2. So it's going to be somewhere. To be somewhere here. So to be somewhere. Yeah. So that's that for then 6, 8 centimeter 6, 8 centimeter 6, 8 centimeter 6. This is 8 centimeter. This is 6, 8 centimeter. 1, 2, 2 here. This is 6. So you have 12 centimeter 11, 12 centimeter 11, 12 centimeter 11 is going to be here. 12 centimeter 11. This is 10. This is 1, 2. This is 11. So 12 each and every. So 16 centimeter 14. 16 centimeter 14. This is 16 centimeter. The 16 centimeter. The 14 is 15 here. 14 will have to be space. So let's see 18 centimeter. 18 centimeter. There's no 18 centimeter. 20 17. 20 centimeter and 17. 20 centimeter 17. This is 20 towards the edge here. Then 17. This is 15. This is 15. This one is 16. This one is 17. This is 17 here. It's 17 here. Okay. So then 22, 24, 22, 24, 22, 24, 22, 24, this 20, this 21, this 22. So do something. So now let's join these together. Remember, I said I'm going to use broken. And I think this 1, 2, and 3 falling. These 3 and 4 not falling. These 3 and 4 will falling. These 3 and 4. So you have this. So that's that. That is that. Yes. All right. So that is the graph for the distance, this particular investigation that has to do with diffusion.
A lot, and you're like, okay, do you have a private tuition? Don't you have? Do you have? And yes, Agile Tech do have a private tuition. So by my right, you can see that we have for the Edexcel and Cambridge IGCSE, and yes, we do have for all subjects. Okay, all subjects. Okay, the popular ones that's always been in demand is mathematics, English language as a second language. Okay, we have physics, we have biology, we have chemistry, and we have the flagship, which is ICT, just here and there. We also have business studies. So if you're interested for a private tuition, I think all you have to do is send an email to the sales team at agiletech@gmail.com, and they would reply to you, talk about your time zone, talk about and the fees really quite affordable. Really, it's really quite affordable. It's when you look at it across other tutoring service, I think this is the one that's quite affordable and you get the very best of service. We also have for the E levels as well, and also for the Edexcel. So we have for the Cambridge E levels and Edexcel E levels. Okay, and we have also across the subject mathematics, which is usually the hot cake when it comes to E level, mathematics and chemistry, and then biology. These are usually one of the ones we have in demand always, all the time. Even physics, for E level. So all you have to do, if you're watching this right now and you are interested for a private tuition, then you can do yourself a solid, okay, and send an email to the sales team at ajiro@gmail.com and they'll reply to you.
B. Explain the effect that increasing the number of drops of ammonia solution has on the time taking this paper change color. Now, as I've said, that ammonia is a base that changes the color of red litmus. So increasing the number of drops, meaning that it means that we are increasing the concentration of ammonia. So the kind of effect it's going to have on the paper is that it's going to change the time quickly, meaning that the time taken for the red litmus paper to change to blue will be shorter because we are increasing the concentration. Explain the effect that increasing the number of drops. So if we increase it more than 3 drops, then that means the time taken for litmus paper to change from red to blue is shorter. So if before, let me scroll up to the table, if before at 4 centimeter distance, we have it takes 2 seconds to reach the 4 centimeter mark of red, damp red paper. It takes lesser than that. It takes 1 second or microseconds color to change from red to blue. That is that will be the implication of increasing concentration for. So looking at this, we can say that this will reduce the timing. It will reduce the time because we should explain it. So the effect will be the timing. The timing will be say timing will be time it takes touching to time takes change then and say that the molecule will diffuse fast. Yes, of course, increase in concentration is to increase in the kind. And when we have fewer drops of say that molecule over is faster. Yes, so that's what. Let's move to question 2B. Question 2B. For 1 drop of ammonia solution, calculate the rate of solution of ammonia between the litmus paper centimeter 24 centimeters. Now, something very important you need to take note, and that is mean rate. There is a formula getting. So we are going to follow that formula. Formula for calculation is let's say mean rate rate of diversion equals to distance traveled divided by time taken. So that is the formula. Though the mean, if we are to follow the normal statistic, mean means average, but this calculation it doesn't mean something different. And because it has to do with you have to use this formula. This may sound as if we are average. This is not. We want to calculate mean rate of diversion. So now we have been given the distance in the question. We have been given 2 distances, centimeter, and don't forget we are trying to calculate distance traveled by ammonia centimeter. So mind you, the diffusion then starts at 4 centimeter exactly. So we have to do is subtraction. So for the distance now, distance, subtraction. And subtraction of what? Say 24 centimeters. So we are going to do this subtraction in the sense that we want to distance traveled by from 4 centimeter mark. We want to know the actual distance. We have to do subtraction, and this will give us. So the distance we'll be using in our calculation is 20 centimeters. We will also do the same thing to the. So how are we going to get the time? Get the time from the table. And let's go back to the table so that we can. Don't forget, we are calculating for one drop. Not. So let's go back to the table. Let's check value, time, 4 and distance. So let's go back to the table. Now, for 4 for one drop, for 4 cm, it is 8 seconds. And for 24, this is 24 here, 38. So the same thing that we have done, distance, we are going to time as. Let's go back. Going to do for distance. For time, that will be 38 seconds minus 8 seconds. 38 seconds. Check and then. That's very good. That's correct. So 38 seconds minus 8 seconds is 30 seconds. So that's 38 minutes. So don't forget the formula. Since we have gotten our value, let's follow the formula. Let's substitute. Distance is 20 centimeter. That's 20 centimeter divided by 30. I'm taking this 30 seconds. And don't forget, according to the question says we should give our answer centimeter per second. So if we do the division, let's press our calculator. That's 20 divided by 30. 20 divided by 30. Going to get 0.66. And according to the question, they say we should maybe round up to smart this. So let's try and verify. If they are silent about, that means we are free, right? 0.66 without rounding up. So the answer is 0.66 centimeter per second. Say 67. Also say 66. So let me just put 6 centimeter. So that is that about question 2B Roman. So let's go to question 2B 5. Question 2B Roman figure 5 says that state how the teacher could improve the ability their investigation. State how the teacher. If a question sounds like this, especially when it has to do with investigation, the one of the things that can be done, the ability of that investigation is to repetition of investigation strengthens. So it means that the more you go, just like a saying that practice makes perfect. So the more you repeat a particular investigation, the more it becomes reliable. Imagine if this investigation is repeated 5 times. No matter the error, if you have little error in the first trial, we may not have it in the second trial, we may not have it in the fourth trial. So if there is an error in first trial, the other ones might not be able to have error. So there is probability of having a more reliable investigation when an investigation is done. So one of the things that the teacher could do to investigation is to repeat the investigation. So the teacher could repeat the investigation. That is what we want to write. Teacher could repeat the investigation. Teacher is teacher could, and you know, using one setup, repeating it might be different. Yes, repeating investigation using just a single setup might be. So another way in which teacher could still use to improve is to use more tools. Yes. So this is just only one setup. This setup. Yes, this setup here is just only one setup. Just only one. So the teacher may have about 5 setups and record table for each setup and have a table like this, a table like this for setup, table like this for each setup. So meaning we are going to have 5 tables for one table for each setup, taking 5 tables. So to increase the reliability of, so just say the teacher could use more tools. Teacher more tools, have more setups, have more setups. So question 2C says describe how the teacher could modify their method to investigate the effect on the education. Okay, so this question is asking the teacher to change the independent variable concentration of ammonia. So meaning that the number of drops will be scrapped. So meaning that the teacher is not trying to vary the concentration, trying to look at concentration of the reaction. The teacher, so temperature will now be so you can say the teacher will have to have 2 different temperatures, one temperature representing high temperature and the other temperature representing temperature. So the teacher will have 2 teacher 2 different temperature values. One 20 high temperature, and the other so apart from that, if you look at the setup, very discover that if the teacher is working effect of temperature, the teacher will need something to maintain high temperature at high temperature and low temperature at. If not, the temperature will be flowing, and that may affect teacher's investigation. Effect of temperature on the reaction. So what it will do will be to use laboratory equipment that will help him to, and the laboratory equipment that you can use to maintain that temperature is thermostatically controlled. water bath that uses a thermostat. So the teacher could also that as well. Say that teacher also water bath. Teacher could also use water bath, regulate its temperature. Yes. So that is that. That is that. So these are what the teacher could do to find method to on the way. So let's move to the. Okay. Yeah, this is another investigation question. This investigation is not so let's take it one step at a time. Let's take it step by step. So we'll be able to understand. Photosynthesis is the process green plants use to convert light energy. So a student uses this method to show that is needed. Step 1. Place a plant in darkness. Place a plant in darkness. So let me be breaking it down before all the way to get to step C. Step 1. Place a plant in. Why are we doing? We are doing that because we want to carry out an and the independent variable according to the way I'm saying the student uses this to show that carbon dioxide is needed. So meaning that we vary carbon dioxide concentration. Either we look at the presence of that absence of carbon. So what we are looking at is actually the effect of carbon dioxide rate of photosynthesis. So meaning that carbon dioxide is going to be our indicator. So the reason why we are placing plants in darkness for is to discharge every starch that has been produced in that plant. Or because we want to embark in a particular investigation. So it's just like want to use a particular electrical. It is common if you want to use a particular electrical app. It's common. Check most manual. They will ask you. They will tell you that you should wait for at least 5 to 10 minutes after it has been plugged electrical source, so that every necessary voltage or surge you have to give, or you can open that electrical app. It's common in want to repair TV, to open it, data or whatsoever. Anything that uses electricity is common. Wait after you have unplugged. Wait for like 5 to 10 minutes. This is because there are components that store charges. So immediately you unplug, there will still be some charges left. So you have to wait till they are completely discharged before you be free to open that electric. Same thing is applicable to. You want to prove that the presence of carbon dioxide makes it possible for plants to photosynthesize. So any plant that you want to use, you have to wait. You have to wait for the plant to discharge itself of all the starch that it has produced. So best method that you will use to wait is by keeping plants in darkness, so that the plant will not be exposed to light. Because if the plant is exposed to light, the plant will continue to. and starch will continue to be produced. So the bait for the whole starch in the plant to use up is for you to keep the plant in sunlight for 48 hours. So by the time you keep the plant away from sun and you take out the plant has already used all the starch in its entire body parts. The starch has been used up for other processes. Respiration. So the starch produced has been used up for cellular activity. So meaning the plant has been destarched. That is the method. That is the term that is used to represent this. What do you call it? Starching. Starching. Pile of starch. So that is what step 1 is trying to achieve. Now let's look at step 2. Step 2 now says remove a leaf and test it first. Meaning that you want to see whether the destarching method has actually taken place. So this step 2, the result must be that it must test negative. Test negative. If the result of step 2 test positive, it means that maybe you didn't discharge properly, or you did the environment where the plant affects. Maybe there's still. That is the reason why we are because the result of step 2 should be that the plant has been really. So let's look at step 3. Step 3 now says enclose one leaf still attached to the plant, a flask containing a bag of solid sodium hydroxide. Now, this is the point where we are taking into consideration carbon. So this particular step, we are taking this step to remove carbon dioxide. Understand? To remove CO. That is the reason why we think this sodium hydroxide. Sodium hydroxide absorbs to remove carbon. So the reason why you are enclosing in a flask is to avoid fresh carbon dioxide from the air come in contact and because immediately the leaf perceives carbon dioxide will start to diffuse into the dark reaction stage of photosynthesis, which is a carbon fixation stage. So the reason why we are enclosing that leaf is to cut off type of environmental supply of carbon. That's the reason why we are keeping it in. And the reason why we are adding sodium hydroxide, despite the fact that kept in it, is that the flask is not a vacuum. There is air in that flask. And remember, air is a mixture of gases. So since air is a mixture of gases, carbon dioxide is also present. So the carbon dioxide that is present in the empty flask where you put that carbon dioxide will have to be absorbed by sodium. That is the essence of absorbed carbon that is already present in the flask, so that you have to eliminate any form of. So that is the reason why we are taking step 3, cut off the sun. Because to be sure that okay, so let's see if photosynthesis will be able to take, despite the fact that cut off the sun. Let's look at step 4. Step 4 says place the plant in bright light. So this is where we will now want to test whether synthesis works after we have. So placing the plant in bright sunlight, giving the plant the opportunity to. That is the test. So step 5 now say test the leaf in the flask starch. So after we have placed the plant bright sunlight for 24 hours, after the 24 hours, go back and remove that leaf that is inside the flask. That leaf, and we test the leaf for starch. The reason why we are testing the leaf for starch is only evidence interesting if starch is present, that leaf as truly. And don't forget, one element or one factor that is needed for photosynthesis to take, that is carbon. Step 6, test another leaf on the same plant. You know, it is only one leaf that we put inside flask and then we introduce sodium hydroxide. The other leaves are still there. So we now have, like, let's say the second leaf will now be like control experiment. So do you understand now? Like a control experiment that let's check whether this leaf has truly photosynthesized or the absence of carbon dioxide has cut off photosynthesis. So the other leaf from the same plant to us whether carbon dioxide that we cut off is having effect on the rate of photosynthesis. So that is the reason why test another leaf. And I believe I've done just that investigation. So the next thing will be to answer the question. Now, question 3A, Roman 1 states that explain why a leaf is removed from the and tested for starch. Let's look at remove a leaf and test it first. Now, the reason why we are doing that is to show that no starch is present. To be double sure that no starch is present. You understand? To be sure that no starch is present. Sure that no starch is present. So that is the reason why I carried out that step 2. And we are also carrying it out to make it a fair test. Yes, we are carrying out that investigation to make it a fair test, to make it a fair test. So those are the reasons why we need to carry out. So let's go to the next question. Let's go to the next question. Question 3A, Roman 2. Explain the expected result of starch test, step 5. So what is the expected, what result are we expecting, step 5? What result? Let's go back to step 5 and let's look at it. Test the leaf in the flask starch. We are expecting a negative result. You know, if the leaves in the flask, in that flask that contains sodium hydroxide, if it tests positive for starch, it means two things: whether the sodium hydroxide has expired, the sodium hydroxide use, whether it has expired, or we didn't rubber bung the flask properly, that air is still entering. And that is the reason why in most of these investigations they will say apply grease or Vaseline around the rubber band, so that the grease or the Vaseline completely prevents the infiltration of air into the flask that contains the sodium hydroxide. So it is either the sodium hydroxide is no longer potent, has expired, or the flask is leaking. You understand? So if the leaf in that flask tests positive, so what we are expecting is negative, and that is what we are right as that. Because they said explain the expected result. So the expected result is since CO2 has been cut off, since CO2 has been cut off, therefore Therefore, no photosynthesis, no photosynthesis has no photosynthesis is taking no photosynthesis. So therefore, no starch is formed. No starch is formed. So how are we going to know that the starch is not formed or the test is negative? So the iodine solution change the iodine, so the color of the iodine solution remains yellow. Now the iodine solution remains yellow. No color change is observed. No color change of the iodine is observed because positive tests show that the color of the iodine on the leaf changes from yellow or brown to purple. That's a positive test. It shows that starch is actually. Let's move to the next. Light is also needed for photosynthesis. That's question 3A, Roman. Question 3A, Roman, says that light is also needed for photosynthesis. Describe how the student's method modified show that light is needed. That means we want to change carbon dioxide concentration, or to show that carbon dioxide is needed. This is to show that light is needed. So how are we going to modify the investigation to show that light is actually needed for photosynthesis? So this is where we are going to make adjustments. This is where we are going to. You see this place? You see step 3. We are going to change step 3. We are going to change step. We are going to remove. We are not going to put the leaf in a flask, because if we do that, flask is transparent. Light can easily penetrate. So even if we put that leaf inside the flask, seal everywhere, if we still carry out photosynthesis, light will still penetrate. If we still have access to light. So what we need to do is to replace step 3 with covering the leaf. Covering a leaf with a black paper or black cardboard. We cover the leaf with black cardboard, and then apply a clip the cardboard together, black cardboard together. Why black? Why not other color? It is because black repel light. Do you understand? Black does not allow light to penetrate or to be absorbed. So black color reflect bounces back light rays. So it won't allow light to penetrate. So that's the reason why we are using black. We can use black nylon. We can use black paper, black cardboard. Show that in order for light not to be able to reach that particular leaf, and then we can use a clip, paper clip, to hold the cardboard in place, to attach the cardboard onto the surface of the leaf, so that it will not fall off, so that the leaf will not be exposed. So that is what we can do to prevent the leaf from being exposed to light. And then we carry out the plant. Then we continue with the way it is structured. The rest of the investigation is structured. It's only step 3 that we are going to change. It's only step 3 that we are going to change. And what we are going to change in step 3, I've said it, I've explained it, that I have to apply, we have to cover the leaf with a black cardboard and attach a clip to hold the leaf together with cardboard firmly. So the students attach black paper, or cardboard to a leaf, and hold them together with a clip. Hold them together with a clip. Yes, hold them together with a clip. Hold them together with a clip. Yes, and expose to sunlight for 24 hours. And expose to sunlight for 24 hours. Expose to sunlight for 24 hours. So the student will attach black paper or cardboard to a leaf and hold them together with a clip, exposed to sunlight for 24 hours. Yes, perfect. So this is what the student could do. Yes. So, and the student should also allow the leaf to have access to carbon dioxide. Yes. The student should allow the plants to have access to carbon dioxide. Or we can say, let me change this, access to expose. Plants, the student should allow the plants to be exposed to carbon dioxide. Exposed to carbon dioxide. So if the student should do that, then and then follow step 4 down to step 6. Follow step 4 down to step 6. So by the time the students place the plant in the sunlight, don't forget that particular leaf will not test positive for starch, because light doesn't reach that particular leaf. So that leaf will not test positive starch, or to test negative. That's one way. Another way in which the students will still carry out this investigation to prove that light is necessary, it's needed for photosynthesis, is to have a 2-potted plant. A potted plant is a plant that is planted in a pot. So, and the reason why we use potted plants for this investigation of photosynthesis is because it is mobile. You can easily carry the plants anywhere you want to carry it to. The plant is planted inside a pot. The pot is packed with soil, and the pot is watered, and the necessary nutrients is present. So the plant is actually growing well. So you can have 2 potted plants, label one as being placed in the dark and label the other as being placed in the light. So instead of attaching paper onto one leaf, just carry the entire potted plants A. You can name them A and B. Carry potted plant A, put it in the dark cupboard. Potted plant B, put it in the light for 24. Do that for 24 hours. And then after 24 hours, bring out that potted plant that you have kept in the dark. Then test it for the presence of starch. Get a leaf from the one that you have exposed to light for 24 hours. Test it for the presence of starch, and then see the results. The one you kept in the dark for 24 hours will test negative. The ones you exposed to light for 24 hours will test positive for starch. So let's move to the next question. That's question 3b. Plants produce carbon dioxide, carbohydrates. Plants produce carbohydrates using photosynthesis. Plants produce carbohydrates using photosynthesis. Explain why mineral ions are also required for healthy to grow. Okay, plants produce carbohydrates using photosynthesis. Carbohydrate is made up of carbon, hydrogen, oxygen. These are the only three elements that is present in. And starch is the product of photosynthesis, and starch is a carbohydrate, meaning starch can only offer carbon, hydrogen, and oxygen to. Starch starch in plants serve as energy store. Energy store. It serve as energy store. So whatsoever thing energy can be used to do is what starch will be used for. But there are some things that starch cannot be used for in the plant, that they can only be gotten from the mineral ions. Yes, they can only be, derived from mineral ions. It's just like saying because starchy foods are energy giving foods, because carbohydrates is energy giving food. Now, I want to relate the matter to human body so that you'll be able to understand. Now, because carbohydrates is called energy giving food, and you want to have energy, you are now eating only carbohydrates, only foods that are rich in carbohydrates. No protein, no fat, no vitamins, no minerals. You are only eating carbohydrates. You are going to experience a lot of nutritional deficiencies. A lot of nutritional deficiencies. You are going to be losing muscle mass because you are not eating protein. So, muscle building, carbohydrate cannot address that. You cannot be eating carbohydrate and expect to have muscle. It's only when you eat protein that you acquire muscle, because protein is a muscle, is a bodybuilding food. It's a bodybuilding food. So that is what they are also saying in plants as well. Plants cannot just be having access to starch every time and expect the plant to function properly. What happens to other aspects of the plant? Do you understand now? So that is where mineral ions come in. So they now said, explain why do plants require mineral ion for healthy growth and for healthy functioning? Now, what you need to do to answer this question is go into the importance of mineral ions, functions of mineral ions, what mineral ions can be used for, the role of the mineral ions in plants. That is what they need. That is what this question wants from you. So now let's start. One, magnesium ion is used in the production of chlorophyll. Yes. So magnesium ion is very important in photosynthesis because it helps in the production of chlorophyll. So let's say magnesium ion helps in the production of the chlorophyll. So it is needed. It's needed for photosynthesis. Synthesis to take place. It is needed for photosynthesis to take place. That's for magnesium ion. Yes. We can also write for nitrate ion. Now, in plants, nitrate ion is used in the synthesis, is used to synthesize protein, plant protein. And you know that plant protein is important in the production of enzymes in plants. You understand? So nitrate ions help in the synthesis of protein protein in plants. Let's also talk about let's talk about potassium ion. Yes, let's talk about potassium ion. So potassium ion helps in water balance. Yes, potassium ion helps in water balance in plant. Water balance plant. So water balance. All right. So you have been able to explain why mineral ions are also required for healthy roots. So let's go to 3C. The diagram shows a cross section. This is the internal structure of the leaf. This is the internal structure of the leaf. And region A. Region A is made up of 3 layers. This thin layer, this layer here, or this layer here, or this layer here. The name of the layer is called cuticle. That is the name of the layer. Followed by this layer here again. By this layer. This layer is called the upper epidermis. Upper epidermis. Then followed by this large rectangular shaped cell, and it is called the palisade mesophyll cell. Palisade mesophyll cell. So this region A is made up of cuticle, is made up of the upper epidermis, is made up of cuticle is the first one, upper epidermis is the second one, then palisade mesophyll is the third one. So they now said explain, that's question 3C. Question 3C says that explain how region A is adapted for photosynthesis. So if we check this question very well, it's going to be 3 marks because region A is made up of 3 layers. So we are going to explain how each layer is adapted for photosynthesis. So explain region A, cuticle. Now the function of the cuticle is that it helps to conserve water. And why is it that cuticle helps in conserving water? Is because it is waxy in nature. It is waxy in nature. Waxy in the sense that it is very smooth. It's smooth. It is thin, and it is shiny. So it becomes difficult for water to escape through that surface. It's difficult for water to escape through a shiny, smooth surface. So that cuticle, waxy cuticle, so we say waxy cuticle helps in conserving water. So that we just because it's 3 marks, you don't need to write that much. So we just represent each layer. We are going to explain how they are adapted for photosynthesis. And you know that water is also needed for photosynthesis. Do you understand now? Water is needed for photosynthesis. So that is the reason why water conservation is very important in the process of photosynthesis. So upper epidermis, because that upper epidermis is thin and is very close to the surface. Do you understand? So it helps light penetration. It makes light to penetrate more. Do you understand? So upper epidermis is thin and very close to the surface. It is thin and very close to the surface. So it helps more light to penetrate into the leaf. So that's that about upper epidermis. Now let's talk about palisade mesophyll. Let's talk about palisade mesophyll cell. Now palisade mesophyll cell, as you can see, this is palisade mesophyll cell. Now you will see a lot of short oval shaped structure lining up inside the palisade mesophyll. So those structures are chloroplasts. So the palisade mesophyll is loaded with a lot of chloroplasts, and they are closely packed. That allows light to be trapped. It encourages more light to be trapped because of the presence of the more chloroplast. Do you understand? So palisade mesophyll cells are packed with lots of chloroplasts which contain chlorophyll, It helps in trapping light, helps in trapping more light for the synthesis. All right. So that's that about this question. We have answered this question. So let's move to question. Okay. Respiration, question 4a. Respiration is a process that provides energy for cells. Respiration can be aerobic or give word equation for anaerobic respiration. This is now, there are two things involved. There are two things involved here. The first is the type of respiration, which is aerobic respiration. Another thing is the kind of equation, which is word equation. Instead of saying two things, let me say three. The location, plant. The kind of equation, which is word equation. The type of respiration, which is anaerobic respiration. Then the location, which is in the plant. Because you see, these three things can actually change in a question. In fact, I can set another question that is directly opposite this. Do you understand? And the answer will not be the same. Instead of word equation, I can make it to the chemical equation. Instead of anaerobic respiration, I can make it to respiration. Instead of plant, I can make it. You understand now? So that is the pointer in a question, pointing you towards the direction that you should go when you are reading a question. So if the word equation, so meaning that we are not writing chemical equation, it is word equation, and we are writing for anaerobic respiration, and it is a plant. And the equation is very simple. That's glucose. So we will not say plus because it is anaerobic. We will not say plus oxygen. Just draw our product arrow, and then we will write ethanol. Now, ethanol is the product of anaerobic respiration in plants. Ethanol plus carbon dioxide. That's all. That's all you need. This particular question. Glucose will be broken down into ethanol, and that is the product of the anaerobic respiration in plants. So let's move to the next question. 4B. 4B is also another investigation. A scientist uses a gas sensor, a gas sensor, a gas sensor to investigate the carbon dioxide produced by a sample of dry seeds. Sample of dry seeds. And the carbon dioxide produced, meaning that this gas sensor will have to be sensitive to carbon dioxide. The gas that the sensor detects is carbon dioxide produced by a sample of respiring seeds. Respiring seeds. Okay. Produced by a sample of dry seeds. The carbon dioxide produced by a sample of dry seeds. Okay. The sensor measures the concentration of carbon dioxide, like I've said, concentration of the carbon dioxide in parts per million, that is the unit, in the air surrounding the seeds. Okay. So the scientist measures the initial and then final concentration. Anywhere we have initial and final, surely have differences of carbon dioxide for dry seeds, for expiring seeds, and for air-retaining seeds. So meaning that we have three categories. The first one is the dry seeds. Dry seeds, that's the first one. Then respiring seeds is the second category of seeds. Then the third category of seed is or air with no seed. Okay, this one, air with no seed. I think this is a controlled experiment. And this is the table representing this investigation. And as you can see the table, we have three testing materials: dry seeds, respiring seeds, air with no seeds. This happens to be the initial reading for all of them, initial concentration of carbon dioxide in part per million, and this is the final, the initial is the final, is the difference, change in the concentration. You have to subtract both values from one another to get this value. As you can see, 492 minus 490 will give you 2. So this place that is empty, what you are going to fill in is to actually subtract 1108 minus 564. The subtraction here is 4. Follow 1 here, that's 10. Here is also 4. Here is 0. So you have 544. 544. That's what you have there. Okay, let me check again. This 10 is 4. This remains 0. That's 544. So and the rate of carbon dioxide production in parts per million. How are you going to? You're going to divide. You're going to divide it. Then you get your values here. So the question now is: give one biotic variable the scientist should keep investigation. Should keep. Let me check if we have any. If one biotic variable the scientist should keep constant in this investigation. Now, this question is talking about this biotic. This biotic is telling you that that part of the investigation that is living. So the only part of this investigation that is living is a seed. Yes, a seed. So that is the what you are going to write, and not just ordinary seed. You should still add some adjectives to qualify, because give one biotic variable. It's going to be a variable, so you cannot just write a seed. You may put type of seed, type or species of seed. Yes, type or species of seed, or even number of seed, or number of seed. Yes, yes. These are variables that should be kept constant during the course of this investigation. You can also say mass of a seed, number of seed, species of a seed, type of seed, mass of seed. So that is what should be kept constant. So that is what should be kept constant in this course of investigation. So let's go to the next question. Calculate the rate in parts per seconds of carbon dioxide production for the respiring seat. So what you need to do is to subtract rates, calculate the rate in parts per seconds of carbon dioxide production of the respiring seat. Now let's go to the table. Let's look at the respiring seat. This is the respiring seat. This is the respiring seat. This respiring seat. So the value that we are going to use is this. This is the value that we are going to use. So how are you going to calculate? You know, we have already calculated the change to be 544. So what you need to do is to divide it by the time in seconds. Just say 544 divided by 180. This, we divide this with this. Do you understand? Divide this with this. So that's how you get your rate. That's how you get your rate. So that will now give you, let's press our calculator. Let's press our calculator. That's 544 divided by, that will give you 3.02. 3.02. So our answer is 3.02. Yes. Check the units, parts per seconds. So the answer is 3.02. So we are going to put 3.0. Let's just put 3.2. Let me check if they ask us to round up. Okay, it's fine. So is that you put 3.0? Okay, it's fine. It's fine. You may put the 2, you may not put the 2. Anyone, it's fine. So the next question is comment on the scientist method and results. Comment on the scientist method and results. Now, what we need to do is we need to comment and we also need to comment about the results that the scientists got from the investigation. Do you understand? The results that the scientists got from the investigation. Now, let's start with the result first. Looking at this table, you will see that the respiring seeds is having the highest concentration of carbon dioxide. Just that the respiring seeds is having the highest concentration of carbon dioxide. So it's part of the result. It's part of the result. So comment on the scientist method and the result. Let's do the result first before we comment about the method scientists use. And we can get the result from the table. If you look at the difference, you compare the difference between the initial and the final, it's just only 2. For respiring seed is 544. Meaning that respiring seeds produce the highest concentration of carbon dioxide. So let's write that. Do you understand? The respiring seeds the highest change in carbon dioxide concentration, produce the highest change in the carbon dioxide concentration, concentration. Yes, that is one result. Let's still look at the table and let's bring out another comment to write. Now, which one is having the lowest? Which one is having the lowest? Okay, let's go with dry seeds. So, though it is air with no seeds, but that one is serving as our control. The kind of comment we are going to write about that would be that no change is observed. No change is observed. No change is observed. No change in carbon dioxide concentration is observed in air with no seeds. So let's first write that of dry seeds, that low carbon dioxide concentration, low carbon dioxide concentration is recorded. Low carbon dioxide concentration is recorded or produced or produced or produced. Or produced. Sorry. Or produced. Yes. In dry seeds. In dry seeds. Dry seeds. Okay. Another one. Another result, another comment on the result is that no change in carbon dioxide concentration. No change in carbon dioxide concentration. No change in carbon dioxide concentration in air with no seeds. In air with no seeds. Air with no seeds. All right, now let's comment about the method that the teacher, is it the teacher now or the student? Scientist, rather. So let's comment about the method that the scientists use. Okay, let's comment about the results. Now, looking at this investigation, I didn't see anything like temperature. That in this investigation, from the beginning to the end, the scientists did not control the temperature, and that is not good enough. So let's include it in our comment that the temperature is not controlled. Or the temperature at which this investigation is carried out is not stated. The temperature at which this investigation is carried out is not stated. So let's say the scientist failed to control the temperature. The scientist failed to control the temperature. Failed to control the temperature. Yes, he failed to control the temperature. Did he repeat the investigation? I don't think so. He did not repeat the investigation. No repetition. And then, for how many minutes? This 180 seconds. That's for 2 minutes. That time is too short. So the method that he used a very short time. He used a very short time. So the duration, the scientists use the shortest duration. Scientists use shortest duration. The duration used by the scientists, the time or duration used by the scientists is too short. It's too short. 2 minutes. 2 minutes is too short. At least should have used, like, let's say 5 minutes. Should have used at least 5 minutes. So that more data will be collected over that period of time. All right, let's move to the next question. Yeast is a fungus used in the production of bread. State the part of the yeast that is made of chitin. It is the cell wall of the yeast that is made up of chitin. Yes, chitin is a very tough material that can be found at the cell wall of most fungi. So it is actually cell wall. That is a part of the yeast cell that is made up of chitin. So question 5, Roman 5A, Roman 2. State the carbohydrate stored in the cytoplasm of yeast cell. Yes, that is the part of the fungi that fungi used to resemble humans or animal cell, because animal cells stores their carbohydrates as glycogen, while plant stores their carbohydrates as starch or store their sugar as starch. So fungi and animal cell, they store carbohydrates as glycogen. So it is glycogen that is the answer for this. Glycogen. Yes, glycogen. So let's move to the next question. Describe the role of yeast in the production of bread. Yes. So describe the role of yeast in the production of bread. Yeast is a fungus. Yeast is a fungus, and yeast produces a lot of enzymes. So enzymes in yeast, they convert starch that is present in the flour. They convert it into maltose. They convert the starch present in the yeast. They convert it into maltose, and then maltase will now be produced to break down the maltose into glucose, and then glucose will now be fermented. So I'm explaining the series of stages that yeast break down the dough or the flour to the level of glucose, where glucose will now have to enter into anaerobic respiration. Glucose will now become fermented, then carbon dioxide will now be produced. So carbon dioxide will now be trapped within the dough, and then the dough will now be rising. Do you understand? As a result of carbon dioxide gas that has been produced during fermentation, the yeast fermentation. So the way I explain it is the way it will be written. Do you understand? So yeast break down starch in flour to sugar. So we say yeast break down sugar. No, let me use sugar. Let me use starch. Break down starch into maltose. The specific enzyme that does this is amylase. Into maltose through the production of amylase, through using amylase enzyme, then yes, using amylase enzyme. Then maltase, yeast also produce maltase. Maltase is produced to break down maltose maltose into glucose. So then glucose is okay, don't let me use the word oxidized, because if I use the word oxidized, is going to affect the anaerobic condition. So glucose is broken down anaerobically. Glucose is broken down anaerobically. Glucose is broken down anaerobically to produce carbon dioxide. Broken down anaerobically to produce carbon dioxide. So carbon dioxide is now trapped. Carbon dioxide is trapped within the dough. This makes the dough rise. This makes the dough rise. So that is the end. That's the end of that is the end of how the role of the yeast in the production of bread. So please take note of the enzymes that I used here. Let me use red ink. Take note of amylase, and also take note of the product that is formed. Amylase is used to break down starch into maltose. It's used to break down starch into maltose. Maltase is used to break down maltose into glucose. Glucose enter into anaerobic respiration to produce carbon dioxide. Carbon dioxide is trapped. Carbon dioxide is trapped within the dough. This makes the dough rise. That's all. So let's move to the next question. Yogurt is another food. Yogurt is another food that is produced using microorganisms. Yogurt is another food that is produced using microorganisms. Explain the importance of controlling the temperature in different stages of yogurt production. One important process in yogurt production is called pasteurization. Pasteurization is partially exposing milk to heat to destroy bacteria, to destroy microorganisms. So, and then the milk that has been heated, that has been partially heated for a very short period of time at about 70 to 95, expose the milk to around 3 to 5 minutes. The milk. And the reason why you have to cool before you add your culture, to make sure that the temperature of the milk before you add the culture won't kill the culture that you want to add. What is this culture that you want to add? Actually ferment the milk to become yogurt, to form yogurt. They normally call it starter. They normally call that culture starter. So yogurt is very simple to make. It's very, very simple to make. So they asked us to explain the importance of controlling the temperature at 10 stages of yogurt production. So the first stage of yogurt production is, let me change back to blue. So the first stage of yogurt production is pasteurization, where milk is heated. Where milk is heated at 70 to 95 degrees C for about 3 to 5 minutes, which is called pasteurization. pasteurization. This method is named after popular scientist was Pasteur that discovered this process of exposing milk to heat for a very short period of time to kill the microorganism. This pasteurization is actually, this is done to kill any microorganisms. Done to kill any microorganisms. So after pasteurization, you have to cool the heated milk. Cool the heated milk to about maybe 40 to 45 degrees. Yes, 40 to this temperature will not harm any bacteria culture that you are adding. So then after this, then you now add your starter culture. After this, you now add your lactic acid bacteria, or lactobacillus bacteria. So then add lactobacillus. This is the bacteria that will ferment the milk, that will give the milk the right texture, that will give the yogurt the right texture. So add lactobacillus bacteria. Add lactobacillus bacteria. Then you are going to provide the optimum temperature by the optimum temperature for enzyme action. Enzyme action. So by the time you provide the optimum temperature, maybe 30, 38, 39, or even 40, that is the optimum temperature. You maintain that temperature maybe overnight. The following day, the yogurt will be formed. So that's that. Yogurt is not something difficult to make. Okay, so next question is explain the importance of changes in pH during yogurt production. During yogurt production, there are some importance of changing pH during yogurt production. Milk normally has pH of maybe 6.7 to 7, about neutral. So immediately lactic acid bacteria or lactobacillus is added, we discover that the pH will drop from maybe 6.7 to like 6.0 to like 5.8 as a result of the presence of the lactic acid, because yogurt is very rich in lactic acid. As a result of is actually the presence of the lactic acid bacteria or lactobacillus that is present. That's the reason why we call them lactic acid bacteria, because they produce lactic acid. So milk has a pH of 6.7 or 7, which the production of lactic acid by lactobacillus reduces to around let's say 6.0 or 5.8, 6.0 or 5.8, or even more than that, so that the pH will become more acidic. So as a result of this pH drop, makes other bacteria to stop growing. That is the importance. The question says explain the importance. That is the importance. So the drop in pH, the drop in the pH, inhibits the growth of other bacteria. Inhibits the growth of other bacteria. So because spoilage bacteria that will make the yogurt to become spoiled, will not be able to thrive at that range of pH. So that is the essence of change in pH. That's the importance of change in pH in yogurt making. Once the lactobacillus starts producing lactic acid into the yogurt, the pH will drop. Once the pH drops, the drop in the pH will keep other bacteria away, will prevent them from growing inside. So so that is that about question 5C. Now let's go to question 6A. Question 6A, Roman figure 1. This is a food chain from a marine environment. We have phytoplankton, we have krill, we have mackerel, we have cod, we have seal. Okay, that is the end. Which is the secondary consumer in this food chain? Okay, phytoplankton is a producer. Krill is the primary consumer. Mackerel is the secondary consumer. Cod is the tertiary consumer. Then seal is the quaternary consumer. Which is the secondary consumer in the food chain? This is the secondary consumer. So the secondary consumer, C, mackerel. C is the answer. C is the answer. All right. So thank you for following us, and thank you for your patience. If you have any question that you want to ask based on the questions that has been solved, get in contact with Ajiro Tech. And if you would like to book a class with me, you can contact Ajiro Tech. So thank you so much. Bye.